Hey everyone, our project name is Integrating Genomics Data with Cancer Registry Records. My name is Kevin Seng and my partner is Jae-Young Kim. In this project, we aim to integrate two types of data. One is the genomics mutation data, which is experimental data come from next generation sequencing of our patients' samples. And the other is patient data in cancer registry database from CIDEFO Clinical Cancer Genomics Division, which is where I work right now as a clinical cancer research assistant. Jay and I have created a query tool that integrate these two data and allows researchers like myself to answer research questions by correlating patient cancer history with specific gene mutations. So for example, if we want to study BRCA1, which is a very common breast cancer gene, uh, and its specific mutation. With this tool, we are now able to see how many patients in our registry has this specific mutation, and also we can see the distribution of the cancer type of these patients. And so with that, we can then learn the correlation between a specific cancer type and these specific gene mutations. And these two types of data sets we have, the first one is cancer registry data. We have about 28,000 patients in our registry. And in the form of CESV, we have de-identified patient ID, cancer diagnosis and age, and also breast cancer hormone receptor status of our patients. The second type is the genomics mutation data. And these are basically 10 Excel spreadsheets of over 1 million rows of data. This is all of the output of the genomic sequencing run. So we pretty much just grabbed every single variant that we can find and that makes up our genomics data. And some key columns that we're able to pull from this data set is protein variant, transcript variant, classification, and also gene. And we will explain some of these more as we do our live demo. The key connection for the two types of data is through a unique identifier called the lab UPN, which is also our sample ID used in our lab. And in the cancer registry, it has its own column, while for the genomics mutation data, this ID is embedded in the sample's column name. So as we pre-process our two types of data, we've decided to split them into three tables. From the genomics mutation data, we're able to split it into first mutation information data. And this is independent from the patient, and it shows information for the specific mutation, where it's located, what type of mutation it is. And this is independent and allows us to reduce the redundancy that we may create by reducing the amount of rows that we output, as we know many patients can possibly share the same mutation. That way, in order to just reduce the output and make it less of a burden when we do query, we've decided to move this as its own table. And for the other part, we created the patient mutation data. And this stuff is definitely dependent on the patient, and it shows the specific characteristics of the mutation based on that patient's genetic makeup. And these involve kind of classification and some specific statistics on the mutation, which tells us how severe this mutation is for this specific patient. And as for patient registry data, we just left it as is. So with mutation information and patient registry data, we convert them into JSON files and upload it to MongoDB. And for the patient mutation data, we converted it to CSV and upload to MySQL. And so with these two database in hand, we were able to connect with those two with our web application, which is built upon the Flask inner framework. And so as we do queries, these data, we can convert them to Spark data frame and query the two database that we have. And in turn, we get the response and we can display them as our data into the user interface. And so I'm gonna hand over to Jay to go over the live demo of our project.
Okay, hello, so I'm Jay. And from now, I'm gonna show you our web application. So we used Flask Python package to build this web. And when we run this, it will gonna give us the local local address, and we can access to the web application. And we used ngrok package. With this ngrok package, we can make a tunnel from this local host to the actual web page. So with this address, while I'm running this code, you can access to our web application. So I'm gonna start in this new window. Okay, so this is our web application. So in here, mainly we have three queries. So patient query, mutation information query, and variant query. So I'm gonna start from the patient query. So in patient query, we can make a query with this lab UPN, which is the ID of patient. So we can get the list of the mutation and the list of cancer history of this patient. And since we have tons of data, it takes a little time to get the result. Yeah, let's wait a second, and it will show the result. Okay. Yeah, finally we have it. So in here you see here's the patient information, cancer history, classification count, and mutation list. So this is the information about the patient. So it is from the mutation, it is from the patient registry data. And here you see the cancer history. So in this case, this patient has breast cancer from the age of 51. So some of the patients has a single cancer history, and some others have multiple of them. So, and then we can see the classification count and mutation list. So I'm gonna tell you about the mutation list first. So if you see here, this patient has thousands of mutations. So in here is the data, the information about the mutation, which is dependent on the patient. But if we click here, it will get the data of the mutation, which is independent from the patient. So I will show you later. And here is the classification count. So you see here's the classification. So it tells us whether these mutations are pathogenic, harmful, or not. So we counted this classification by using Spark data frame. So you see here, in this case, this patient has 101 pathogenic mutations. And if you click this, it will gonna give us the information about the mutation, which is independent from the patient. So let's wait a minute. Yep. So we have it. So this is the information about the mutation, and you see here's gene, region, symbol, position, this kind of things, and you see here transcript ID and transcript variant, and these can be multiple. There can be multiple results, so it is expressed as a list. So let's go back, and this is the mutation information query, and it will show the page similar to what I just showed you. So this is the mutation ID, and it will show the result. Yeah, this is it. And let's go back. And finally, we have variant query, and this is highlight of our web application. So you see here's 4 slots: protein variant, transcript ID, transcript variant, and gene. So we can make a query with these all 4 of them, or just query with some of them. So I'm gonna make a query with these 3.
And so for more information, protein variant, transcript variants, and gene, these are basically addresses of these mutation in this specific gene. For protein variant is the mutation in the protein form, and for transcript variant is the mutation in the DNA form.
Okay, thank you, Kevin. Yeah, because we have tons of data, it takes time. Yeah, finally we have it. So this is the information of the query we made, and here's the number. So this number of mutation and the number of patient means there are 6 mutations which has all these protein variant, transcript ID, transcript variant, and gene, and there are 5 patients who has all of these. So, and here we have the classification count. So it is similar to what we saw on the patient query. So we have 6 uncertain significance mutations. And this is the mutation, which has all of these. And here's the cancer type count, which is the most important thing in our project. So you see here, there are 4 breast cancer and 1 thyroid and 1 uterine cancer. So it shows the distribution of the cancer type of the patients. So by knowing the correlation between the cancer type and the protein variant, we can plan the preventative treatment of the cancer. So this is real life applicable. So this is one of the unique feature of our project. And below here, you see the mutation list and the patient information. So these are the information of these mutations and patients. So we have only 5 to 6 patients and mutations in this query because we made pretty much specific query. But if we make like this, only make a broad query with only gene, and we can get thousands of results. So it is, yep. Let's see the results briefly.
And this allows for great flexibility in our query tool based on the liking of the researcher. If he or she wants to just study this gene, MutYH, and see what kind of results we have, they can do this query we're about to show. Or if they come across a new paper and find a new studied variant, they can then be a little bit more selective and search by gene and protein variant, etc., etc. So this just gives us a lot of flexibility in our research.
Okay, so you see here are thousands of mutations and classification and patients. So yeah, this is very applicable to real life. And yep, this is all for our project. And thank you for listening. Thank you.
Thank you.